occur clearly in flies.
Okay.
So,
thank you, Dr. Badmos.
thank you.
I would like to request all the participants to discuss the remaining questions on Slack. Our next speaker is Dr. Katja Röper from the famous Laboratory of Molecular Biology, LMB, University of Cambridge. She is a special collaborator to DrosAfrica, and today she will be talking about mechanisms and mechanics of tube morphogenesis. To you, Dr. Röper.
Hello, whilst I share my screen, thanks first of all to Isa and the other organizers for inviting me to talk. It's a really cool meeting. So I will tell you, I hope this is now. Yes, I hope you can all see my presentation now. So my lab is interested in, wants to study the dynamic behavior of epithelial tissues during organ formation. And basically what we're interested in kind of ties in very nicely with a lot of the things that Eric Wieschaus told you all about yesterday afternoon, very beautiful in his talk. Because what we want to understand epithelia, because really at the beginning of development, most animals start off as a simple structure, very often a simple polarized epithelial layer. And this is true for us humans, but also for most of the classic model systems that are used, such as mouse, zebrafish, or the fly. And then these simple structures, simple epithelial sheets undergo complex processes of organ formation during embryonic development over differing time scales, depending on the organism, to end up in these beautiful embryos with beautiful structures highlighted here. And so what my lab in particular wants to understand is how organ shape is encoded by the genome. So the initial epithelium, for instance, of a fly embryo shown here is very uniform when we look at the shapes of cells. So you see nuclei here at the moment. But then this embryo is already very quickly diversified transcriptionally. And these transcriptional changes that we can see and the patterning will define embryonic regions and organ primordia that then are turned into physical changes at the molecular, at the cell and tissue scale to drive organ formation over time. So we want to understand the link really between the transcriptional changes and the physical changes that drive the formation of organs in the embryo. And we're particularly interested in the formation of tubular tissues from epithelial sheets because most, for instance, of our own internal organ systems are tubular in epithelial nature, such as lung, mammary gland, or kidney. And the same is true, as you will be aware, of the fly as well. So the organ system we study a lot in the lab are the salivary glands in the fly embryo. And the tubes of the salivary glands form from an initially flat epithelial sheet with not many distinguishing features shown here. But again, if we look at transcription of key factors, then the primordium of the tube of the salivary gland here in magenta is already defined at this stage, where at the epithelial cell shape level, we can't really see very much yet. And then over only a few hours, this flat patch of cells transforms into this tube through a so-called budding process. So the tubes of the salivary glands, the primordia from which the tubes form, so-called salivary gland placodes, are specified on this ventral side of an embryo. So if you look at the ventral side, there are two patches of about 100 cells on either side of this ventral midline. And a very common morphogenetic process involved in the formation from tubes from flat epithelial sheets and also in the tissue bending that we see here is the constriction that initiates the formation of these tubes here. So if we look in surface views here or cross-section views of the forming organ, then you can see that cells in a corner of this primordium, in this dorsal posterior corner, seem to start to constrict. This constriction spreads out across the tissue and cells disappear into the inside. And if you look in the cross-section, you can see how the tissue starts to bend and this tube starts to form and stretches further and further into the embryo. So this is a very nice tractable model process of tube budding because it's accessible to live imaging and all the genetic approaches that we have in flies, but also because from the starting point of this flat, not very interesting yet epithelial sheet to the formed tube, there's basically no cell death or cell division occurring, which means that really going from here to here, the whole process is driven from start to finish by changes in cell shapes and cell rearrangements. And so this being Drosophila, as we already just saw also in the last talk, we can very nicely image these processes and then get and quantitatively analyze what's going on. So I'm just showing you a movie of this process in the surface and cross-section views, where you can see that it's really a combination of changes to individual cells in this primordium, but also the coordination of changes of many cells that drive the formation of this tube from the primordium. So we know, and you heard about that from Eric yesterday, that apical constriction of cells, which, as he spoke about, drives mesoderm invagination, for instance, is an important process that can drive tissue bending. And we already knew in our tissue primordium that, again, myosin contraction and concentration apically in a certain patch of cells is what initiates the tissue bending also here. And we showed a number of years back that again, it is a very dynamic pulsatile pool of apical medial actomyosin in these cells that goes through cycles of increase in intensity and decrease. It's very motile that drives this constriction apically. We wanted to understand better what really drives this formation of a narrow tube that is important for the final function of the organ. And that is not only true in the salivary glands, but in many other tubular organs. The kind of final shape of the organ will be very important for the proper function of the tissue later on. And so what we did a number of years back, and I will just summarize this because it's published, but it's the basis of some of the other things I want to show you today, is we took a postdoc in my lab, Yara Sanchez-Corrales, together with Guy Blanchard at the University of Cambridge here, utilized and developed quantitative so-called morphometric methods that allowed us to dissect this process in more detail. So we took time-lapse movies of embryos, and after accounting for the overall curvature of the surface of the embryo, we could then identify and segment individual cells, and that allowed us to track these over time and look at their behavior and changes, and then perform so-called strain rate or deformation rate analysis. And so this strain rate analysis basically analyzes what is the overall shape in the tissue shape change that we can see. How does the shape of the patch of cells that we're interested in change over time? And we know that this is basically coming from two components, from changes of the cell shapes of individual cells, and also changes of neighbor connectivity of these cells, so-called cell intercalations. And in most organs, this will also have a contribution from cell divisions, generating more cells, or cell death or cell delamination. But as I told you a couple of slides back, we know that in our tissue primordium for the salivary glands, there is no cell death or cell division occurring. So really, all shape at the tissue level should be accounted for by a combination of these two. And so from the movies we can quantify the overall change in tissue shape. We can quantify the overall change in shape of individual cells, looking at small domains of cells, small groups. And then we can deduce from that how much cell intercalation must really have occurred at between any given time points, because those two together should add up to the overall tissue shape change. And so by doing this, what we could discover was that in this initial patch of cells, these 100 cells that will form a tube over a few hours, we could see that there are two dominant behaviors in different regions. So near the point where this invagination point in the corner of the tissue will form where the cells might dive into the embryo to form the tube, the cells constrict apically through this isotropic constriction. But further away, the cells early on do not at all change their apical area. Instead, they show very active intercalation behavior, which basically leads to a contraction of the tissue circumferentially and the radial expansion. So cells are fed towards the invagination point continuously. And this really seems to be due to different pools of actomyosin apically in these cells. So near the point of invagination, as I showed you in a movie earlier, we see this very active contractile apical medial actomyosin, but at a distance, we see a strong concentration of actomyosin in circumferential junctions, which help the cell intercalation behavior. So, but we didn't only look kind of apically in these cells in 2D, but we also looked along the whole length of the cell to understand how these rather elongated epithelial cells in total contribute to the formation of the tube. And so there we could see that this apical constriction here, in fact, in 3D is a cell wedging behavior that happens close to where the invagination pit is forming and in the cells that continuously internalize. And that cells across the whole primordium show a tilting of the apical sides towards the point of invagination. And that cells also along their length from apical to basal show behavior that we call interleaving, where they basically change neighbor connectivity along the length of the cell, which also helps the cell intercalations we see and helps to feed cells continuously into the invagination point. So this was interesting. We see this very clear split of behaviors early on. What we wanted to find out now is really, how is this controlled? Is this all through genetic control? Are there mechanical implications of the process that we wanted to understand? And so we knew from the literature that there's a cascade of transcriptional regulation that defines the primordium downstream of, and don't worry about most of the names of factors here, downstream of homeotic genes that specify the identity of the patch of cells. In the next level of transcription factors that are activated, there are two that are called forkhead and hookabine that have been described as possibly being just upstream of factors that could be what we would call morphogenetic effectors, so molecules like myosin that really help to change the shape of cells or might help change neighbor connectivity. So what we were thinking is, and we knew actually from old in situ hybridization studies, that the RNA for these factors is first found where the invagination pit will form, and then seems to over time slightly spread out across the placoid. And there's some other factors that were described to first be initiated near the forming invagination point and then spread out. So what we imagined was that this temporally graded expression of these determining transcription factors that might switch on important targets, and some of the downstream targets might first of all be involved near the forming invagination point to initiate the expression of the apical medial actomyosin, which initiates this apical constriction and lets the pit form. And then this is spreading out and could help to make more and more cells continuously constrict and then invaginate. But such an expansion over time of an expression pattern of a transcription factor could also help to define boundaries across the primordium that could help polarize these junctional myosin accumulations that we need for the initiation of these cell intercalations that we see that help to move cells towards the invagination point. And we know that this then actually combines in a study that we did previously with actually, once the pit has formed, a pulling force from the pit and therefore a mechanical response to these cell intercalations that help to resolve them to feed more cells into the invagination point. But to really see if this upstream path of this temporally graded expression of important transcription factors is correct, we kind of wanted to analyze this in a bit more detail and with better methods than were available. So we used endogenously tagged versions of hookabine, tagged with Venus, or forkhead tagged with GFP, to analyze their expression pattern live across the early stages, or actually across a very long period, including the very early stages of formation of the tube. I should say in any of the plots that have a time stamp, time zero is what we define as, or is the point always at which at the tissue level we can start to see the first tissue bending or invagination forming. So there are lots of cell behaviors and cell shape changes that happen prior to this point, but they do not at the tissue level yet lead to any changes. And so if we now analyze the intensity of hookabine and forkhead across the tissue, if I just summarize that, what we can see is, and so in this plot, we have at the bottom here the radial distance from the invagination point in the corner of the primordium, this is called pit, which is on the right here. And then intensity, and we have here three different time points. Basically, we can see that for both hookabine and forkhead, the expression really starts very strongly in blue here near the forming invagination point, and then slowly spreads out further and further anteriorly across this placoid. So this temporally graded expression of these two upstream factors is definitely happening. But what could be downstream? And in the literature, there was a good indication, again, of a factor you will have heard of yesterday from Eric, which is a gene called FOG for folded gastrulation. And FOG includes this ligand to a G protein coupled receptor that downstream through a very short cascade through this protein concertina G protein activates through Rho GEF and Rho G protein GTPase, activates Rho kinase, which then will lead to activation of myosin in these cells. So FOG is a good target that could directly induce myosin activity in the cells. And if we look at FOG levels using an antibody against the protein and compare it to the apical cell area in the region of our placoid, then there's a very good correlation always across different, so before invagination, at the beginning of invagination of these two placoids here, or during invagination, a very good positive correlation of the amount of medial myosin that we can see and the medial FOG intensity. And wherever FOG is high, the cell area is decreasing. So where FOG is high, we see high apical medial myosin and the cells constrict, leading to invagination. But so how does the myosin look if we look at this in more detail, actually, at these different time points? So this is now looking at endogenous tagged myosin in these embryos. And if we look in the region here, on the down here, where the invagination point will form, then in addition to junction myosin, we see lots of foci of apical medial myosin that, if you look in movies, show this dynamic behavior here. Whereas further away from the pit, as I said before, we just see this junction accumulation that leads to cell intercalations. But now with time and with FOCAT and Huckabine expression spreading out across the placoid and FOG increasing in these cells, we now see more apical medial myosin always ahead of this invagination point. So here now you can see the hole through which some of the cells have already invaginated into the embryo. And even further away from the invagination point, we now start to see the appearance of small clusters of medial myosin. So if we quantify this from many different movies and plot it over time, so here on the y-axis is against time zero is when the tissue bending starts, and this covers about 80 minutes of the process. And again, this is the distance down here with the invagination point at the beginning of the movies on the right-hand side here, the pit. Then you can see that the highest intensity that is plotted here of medial myosin in green is always tracking right in front of the invagination point. So always the cells that are right in front will switch on very high myosin medial intensity. And another characteristic we can analyze of the myosin is in its dynamic behavior. So it shows these pulsations, these waves of intensity increase and decrease. So we can define the cycle lengths between two peaks. And the faster or the shorter the cycle length is, the more strength this myosin supposedly has to contract cells. And so again, we can see the shortest cycle length also tracks here right in front of the edge of the pit over time. So what we think this means is that really once invagination has initiated on constriction and the pit is forming, we have what we would kind of call a standing wave of apical medial myosin activity and apical constriction that tracks in a near fixed position ahead of this invagination point, whilst it moves across the tissue and more cells internalize. So is this now really linked to the patterning of the two transcription factors I described earlier? And to look at that, we now compared in individual movies the intensity first here of Huckabine with the apical area change. So the plot again is time of the invagination here on the left, but now looking a lot before actually tissue bending at zero here starts. And again, positioned across the placoid is plotted on the x-axis, so the pit is on the right. And we see the highest venous Huckabine intensity is at about half an hour before tissue bending starts. And at about 0 minutes when the tissue bending starts is in terms of apical areas when we see very strong constriction. So there's about half-hour delay between the two. And the same actually is true if we look at FOCAT intensity tagged with GFP. Again, at about half an hour before any tissue bending starts, we see 4CAT being very strongly expressed at the position of the pit, and then actually this highest intensity again for 4CAT tracks at a fixed distance to the forming pit and the continuous invagination. And here again, this is with a half-hour delay to the strongest apical area change. And about half an hour would allow enough time for any targets of 4CAT and Huckebein to be expressed, and assuming it is fog, be secreted in the cells and have an effect on the cells from which it is secreted. So therefore, we think there's a good correlation between really this dynamic patterning of these two transcription factors and the standing wave of apical constriction that we see in this placode. So I told you earlier that we don't only see this apical constriction, which I just focused on now in terms of the control of how we drive a continuous invagination through the patterning of these transcription factors, but there's also the cell intercalation that we see in cells further away from the pit earlier on, driven by this junctional myosin sitting there. But what we can see actually is if we now look at just the apical area of cells, either these ones that are from the beginning close to where the invagination will form. So these we always knew from previous work already start to decrease the area. So this is the apical area change that is plotted here over time. They decrease the area continuously over time until they're all internalized. The cells further away, as I told you, they first do not change the area much because they show the cell intercalation, but then from about 20 minutes after the start of invagination, they also start to now constrict and show apical constriction, because by this point now, these cells have internalized and these have reached a position close to the pit. So through this standing wave of apical constriction, they now start to constrict. But this means that really these cells further away seem to switch their behavior from predominantly cell intercalation to predominantly apical constriction once they reach the right position. And in fact, we can see this if we look at individual cells and their behavior across the placard. So again, this is the primordium patch. In pink here are cells defined as being close to the forming invagination points. The gray ones are further away. We now look at individual cell examples. And if we look at the apical area of these cells close to the pit, they immediately start decreasing their apical area. Whereas the cells further away remain with the same apical area for quite a long time before they then also start to constrict very rapidly. And as an example, I've shown you this blue cell here undergoes cell intercalations earlier on before it starts constricting that help this expansion of the tissue towards the invagination point. So this group of cells rearranges over time into this elongated shape that is much more elongated towards the invagination point and helps the cells move into the invagination. So cells switch their behavior from cell intercalation, at least the ones further away, to the apical constriction. But then we wanted to know why actually is the setup in the placode in this way that this invagination point is in the corner, is in an eccentric position? Why is it not symmetrical? Is there a reason for that? So we have in the wild type, what we see is this eccentric asymmetric pit location. But why can't it also work with a central symmetric location? And so we did a thought experiment first to see if we could come up with an explanation that we could then test in the flies why this might be the case. So if we have, as in the wild type, this eccentric small pit that starts to form originally, then over time with a new and a further corona of cells here color-coded invaginating, if we look at the number of cells in these coronae, then although the number of cells increases slightly, it's a very slow and steady increase, and more or less a very similar number of cells invaginates, which probably helps to generate a narrow lumen of a defined diameter that remains like that throughout the process on the inside of the embryo. A central invagination would lead to larger and larger numbers of cells with each corona being internalized. That's this pink curve here. And if we have an enlarged pit, or that covers some peripheral and central areas, then again we have a more steady number of cells invaginating here in gray, but much larger, so it would be a much wider lumen of the final organ. So we wanted to see if we can test this in some way, and actually we can, because a mutant in this transcription factor Huckebein that I just showed you is important for the patterning in the placode. and probably drives the fog expression that drives the apical constriction spreading out across the tissue. In a mutant for this factor, we actually end up with a central constriction and invagination that at later stage, instead of, as in the wild type, we have this very nice narrow lumen of cells, we now have tubes that invaginate, but they have a enlarged, widened sac-like lumen that is very different to the wild type. So we can test this central invagination and it leads to the wrong shape. And we can also test this very large invagination covering peripheral and central areas by overexpressing this ligand fog all across the placard. And if we do this, again, instead of in the wild type here, where we have in a cross-section a very nice circular narrow lumen tube, and here in a different cross-section, when we overexpress fog, we have this very distorted enlarged lumen that forms. And if in these conditions now we count dynamically how many cells invaginate through the forming pit, either in the center or this much larger region, then very similar to our theoretical considerations, yeah, in the wild type, actually, we have a very small number of cells internalizing with each corona of cells that goes in. In the Hookabine mutant, more and more cells internalize with every corona of cells that goes in. And in the fog overexpression, basically most cells have internalized within the first two lots of cells that go in, and we form these big sacs on the inside. So really, to form a regular narrow lumen tube, it seems to be important that we have this eccentric asymmetric setup of the placode with the pit in the corner that allows this continuous internalization of fairly identical numbers of cells. And we think the cell intercalations we see actually also help this continuous supply of new corona of cells in identical numbers in this near to the pit position, where they then switch to apical constriction and drive the invagination. So in summary, I showed you that during tube budding of the salivary gland from a flat epithelium, apical constriction and cell intercalation behaviors combine to drive this continuous internalization. And we see this switch of cell behavior from cell intercalation to apical constriction as cells approach the invagination point. And this switch seems to create a sort of standing wave of apical constriction near the pit. And we think this eccentric setup of the placode is key to really internalize a narrow lumen tube that will be important for the later correct function of the tissue. So just to say again, the two postdocs who did the work, Yara and Guy, and to thank everybody else in my lab and in my institute and people involved in the project. And I thank you for your attention and take any questions.
Thank you very much for the beautiful talk. We have a few questions. I will start with, I have a question myself. Why do only specific, like a specific group of cells, they start to invaginate? They are the ones that show myosin contraction, whereas all the other cells around the periphery, they just elongate and stretch. And the ones in the center, they start the invagination, whereas the surrounding cells, they only stretch.
So the ones that start the invagination are the ones that will be the very first ones to express these two transcription factors, FOCAT and Hookabine. So their kind of overlapping expression in these cells in the corner drives the expression of this ligand, FOG, which then switches on in the cells that express FOG and secrete it. So it's an autocrine signal. In these cells, it switches on myosin activity that drives the apical constriction there. The cells further away, they actually don't stretch very much. They undergo these intercalation and neighbor exchanges. So we don't yet know what is upstream of this junctional myosin polarization in these cells, but we see myosin mostly really in arcs that are circumferential in this tissue, and that helps pull cells together into these rosette structures, of which I showed one, that helps to kind of feed cells continuously towards the invagination.
Okay, thank you. The next question is by Hamid Badbos. He asks if this type of apical constriction is conserved in other epithelium.
Yeah, so apical constriction, I mean, as you probably will have heard from Eric yesterday, so apical constriction is important in a lot of tissues for tissue bending, not only in flies. But in flies, there's some other patches of the epithelium that undergo very similar behaviors. So there's part of the respiratory system, the posterior spiracles, that have a placode at the posterior end of the embryo, which look nearly identical to what we're looking at. Also, the imaginal discs invaginate from the epithelium of the embryo at the end of embryogenesis, and they undergo apical constriction, which looks very similar to what we see here. But also in discs, so in the eye disc, for instance, there's this morphogenetic furrow, which is a kind of row of constricted cells that sweeps across the tissue, behind which the photoreceptors differentiate. That is apical constriction driven by increased myosin apically that drives that. So it's a very common principle in epithelia that tissue bending is driven by apical constriction and myosin apically.
Okay, thank you. The last question we have from Lola. She asks, how is the on-off cycle of myosin regulated? Is myosin assembled into filaments? What changes or the amount of what changes the amount of myosin or both?
Yeah, it's not clear. I mean, I think the best studies for that come from Thomas Lecuis's lab, looking at that actually it's a kind of it's a system that drives its own pulsation, that once the myosin or the regulators have constricted, they tend to switch their own constriction behavior off, and actually myosin pullin and actin filaments can help lead to disassembly of these actin filaments. So it might be that just the network on which the myosin works disassembles once it's constricted enough, and then you start a new cycle because you need to kind of provide new actin filaments that the myosin can work on. We haven't looked at that in detail here, but that's at the moment, I think, the thinking in the field, that because this pulsatile behavior of apical actomyosin, I mean, it's very obvious and prominent in tissues undergoing morphogenesis, but you can actually see it at a very low level in all epithelial cells. So it might be an intrinsic feature of the cortical actomyosin in most cells, but when you want to use it to change shape, you ramp it all up and amplify the system to really achieve a defined change.
Okay, thank you. For all the questions that haven't been answered, please continue these discussions on Slack. The speakers would be more than happy to assist you with your queries there. Thank you, Dr. Röper. The next two talks will be chaired by Amelia.